Today's essential question is, how are traits expressed from DNA? Our objectives are to summarize the events of DNA replication and relate the DNA molecule to chromosome structure. Here are our key terms. Get your notebooks out, be prepared to take some notes, and jot down any questions you have. So think about it. Before a cell divides, its DNA must first be copied. How might the double helix structure of DNA make that possible? Before we complete this lesson, study this diagram for a moment and make a list of questions you have about the diagram. As you read and listen to the lesson, write down the answers to any of the questions you have. In order to understand DNA replication, we should first review for a moment what DNA looks like. Remember that one half of a DNA strand has all of the information needed to build the other half. The matching strands are complementary. Remember that adenine always bonds with thymine and that cytosine always bonds with guanine. Before a cell divides, its DNA must be replicated. Remember that this is happening during interphase of the cell cycle. Replication is the process of copying a cell's DNA. This ensures that each daughter cell will have a complete set of DNA. Let's take a look at the replication process. During DNA replication, the DNA molecule separates into two strands and then produces two new complementary strands, following the base pairing rules. Each strand of the DNA double helix serves as a template or a model for the new strand. The first step in the replication process is that the two strands of the double helix separate or unzip, allowing two replication forks to form. As each new strand forms, new bases are added following the rules of base pairing, A with T, C with G. If the base of an old strand is adenine, then thymine is added to the newly forming strand. Likewise, guanine always pairs with cytosine. The result of replication is two DNA molecules identical to each other and to the original molecule. Each DNA molecule resulting from replication has one original strand and one new strand. The replication of DNA is carried out by a series of enzymes. The enzymes first unzip the molecule by breaking the hydrogen bonds between the base pairs and unwinding the two strands of the molecule. Each strand then serves as a template for the attachment of the bases. The main enzyme involved in DNA replication is called DNA polymerase. DNA polymerase is an enzyme that joins the individual nucleotides to produce a new DNA strand. DNA polymerase also proofreads the new strand, making sure that the new strand is a perfect copy of the original and that there aren't any mistakes. Replication looks a little different in prokaryotic and eukaryotic DNA. In the cells of most prokaryotes, DNA is a circular molecule found in the cytoplasm. Eukaryotic cells, however, have up to 1,000 times more DNA, and nearly all of the DNA is found in the nucleus. Let's review the steps of DNA replication. First, the DNA strands separate or unzip. Two new complementary strands form, following the rules of base pairing. Each original strand ends up with a new complementary strand. Let's remember as well that DNA polymerase is the important enzyme involved in DNA replication. It joins the individual nucleotides to produce a new DNA molecule. It also proofreads the new strand. Let's try a few review questions. The DNA that results from replication consists of two molecules composed of old strands, two molecules composed of new strands, two molecules that each contain one new strand and one original strand, or one molecule with two strands and one molecule with two original strands. If you chose C, you're absolutely correct. The enzyme that joins individual nucleotides to produce a new strand of DNA is called amylase, DNA polymerase, RNA polymerase, or telomerase. The correct answer is B. Let's try one more. Before a cell divides, it duplicates its DNA in a process called base pairing, transformation, duplication, or replication. And the correct answer is replication. So let's review again. Our big idea today was how are traits expressed from DNA? And I think we met our objectives. We summarized the events of DNA replication, and we related the DNA molecule to chromosome structure. Bring any questions you have with you to live class.